He holds a bachelor degree in biotechnology and a master's and doctorate in biological science from the University of Brasília, so it's a person from of university. He's a current postdoctoral researcher in Professor Elizabeth Ferron's laboratory. His primary research focuses on isolating toxins from scorpion and spider venoms to study their effects in voltage-dependent ion channels using electrophysiological techniques. Please welcome Mr. Dr. Diogo Tibery, I guess. Be my guest.
Hello, everyone. And first, I want to thank the organized committee for the invitation. For me, it's very important. It's my first presentation in an international conference, and also it's just 4 months that I have my PhD degree, so it's very important for me. Where is the pointer? So, my presentation is entitled Investigating the Toxicity of the Brazilian Scorpius venoms, focus on ion chain active peptides and their therapeutic potential. So, to start this presentation, it's important to understand how big is the problem of envenomation in Brazil. In the year of 2021, 62% of the accidents were related to scorpions, 10% were related to snakes, and the others, 27%, were related to others, being disorders, bees, wasps, caterpillars. And also, it's really important to study the scorpion venom peptides, looking the increase of the accidents with the scorpions in the last 11 years. As you can see, between 2012 and until 2022, you have an increase of 186% of accidents with scorpions. And also it's important to see where this accidents happens in Brazil. Most of the accidents happen in the southwest part of Brazil and northwest part of Brazil. This is related to the amount of the people that lives in this part of the city. This is the most populated part of Brazil. So you can see a high amount of accidents in states of São Paulo and Minas Gerais, and also in the northwest of Brazil, and some accidents here in the central west. And also possible to see some small case on the south of Brazil and the north part of Brazil. The main symptoms of accidents of envenomation by scorpion stings are pain, and this pain could lead to other symptoms. Could happen other symptoms also, as cardiac arrhythmia, hyper or hypotension, muscle paralysis, and mental confusion. In this map, it's interesting to see which species is related to the accident it's related to the accidents in each region. In the southwest of Brazil, most of the accidents are related to Tichus baiense and Tichus cerulatus, as could be seen here in green for the Tichus baiense, most on the states of São Paulo, a little bit going to the states of Minas Gerais, and Tichus cerulatus in this dark green, in this dark gray, as you can see, this high dispersion from the states of São Paulo, until north and in the south of the northeast of Brazil. So it's one of the species of the most distribution in Brazil. In the northwest of Brazil, the principal culprit of accidents is the Tichus stigmurus, and in the Amazonic region, the culprit of the most of accidents is Tichus obscurus. And also, I would like to highlight the Tichus fasciolatus. It's a scorpion that is found in the rural areas of the Federal District. It's not related to many of accidents, but is one of the scorpions that we study here in the lab in Brasília. So I would like to present also the Tichus fasciolatus. The main culprit of accidents in Brazil is Tichus cerulatus. This is due for two specific regions. Two specific regions. One is the high toxicity of this scorpion, and also the easy to dispersion that this animal has in Brazil. The first point that's important to mention about the scorpion is the method that this scorpion reproduce. It use teletocous parthenogenesis, or this scorpion just has females, and the females just produce more females. It's an all-female brood type of scorpion, and also it's a really opportunistic species. These species take advantage of the human activities that promote some disclimate environments, and this is really good for this animal to disperse to this region and be allocated there. As could be seen here in this map that show the distribution, the initial distribution of the Titus cerulatus that was first identified in the state of the Minas Gerais, specifically in Belo Horizonte. And also after that, we can see that the dispersion went to the southwest of Brazil, to state of São Paulo, also went to the some of these populations went to the northeast of Brazil, and also went to Brasília. In Brasília in specific, it's really interesting to see because Brasília is constructed in the 50s, and also in the first inventory made about the scorpions present in Brasília in the 1975 was not observed any species of Titus cerulatus. Was just observed at that time species of Titus fasciolatus, and some species of genus Ananteris and Broteurus. But in the second inventory made on the 90s, for the first time it was observed the presence of Titus cerulatus. So it's important to see that in just 15 years, the dispersion of this scorpion infestated Brasília. And also, it's in some new reports, it was possible to observe the presence of the Titus cerulatus in the far south of Brazil, in the state of Rio Grande do Sul, and some other reports demonstrate the presence of Titus cerulatus also in Argentina and Chile. When you talk about the scorpion venom composition, we have a lot of different compounds that could be found. For this presentation, I will just focus on the specific toxins that target ion channels. In this transcript analysis of Titus obscurus and Titus cerulatus, it's possible to see that almost 12% of the transcriptomes found are related to toxins active on sodium channels, and 13% is related to toxins active on potassium channels. This is almost the same as in the Titus obscurus, with a little bit of difference, but it's between 10 and 12% of the transcripts. So when you talk about this, the active toxins on sodium channels, we have two big classes. We have the alpha toxins and the beta toxins. These toxins are really similar. Both have the folding of the cysteine stabilizing alpha beta folding, as could be seen here after here. It's possible to see the alpha helix and here the beta structures. Alpha toxins interact in domain 4 of the sodium channels, specifically on the voltage sensors, and this interaction promotes a trap of this voltage sensor in the dial position. This stabilization of the dial position doesn't allow a specific motif named IFM to interact with the inner part of the pore and inactivate the channels. And the other type of toxins, the beta toxins, interact with domain 2, also interacting in the voltage sensor domain. In this case, the beta toxins promotes stabilization of the voltage sensors in the up position. And this stabilization in the up position is essentially promotes facilitates the excitability of the channel. It's also possible to see here in the simulations the interaction of these two types of toxins. This could be seen here: an alpha toxin interacting domain 4 in the alpha helix of the voltage sensor domain, and also in domain 2, it's possible to see the beta toxins interacting with the alpha helix of the voltage sensor domain 2. The other type of the toxins that we have in the scorpion venom peptides of the Titus genus, of the genus Titus, are the toxins active on potassium channels, the KTX. And the KTX are the most diverse one that we can find in the venom, because these toxins have 4 types of different folds. We have the cysteine stabilizing alpha beta, it's the same pattern that we found on alpha and beta toxins active on sodium channels. We have cysteine stabilizing alpha-alpha structures, and also we have a Kunitz type of folding or a ECK type of fold. These toxins, different from the sodium channels, different from the ones active in sodium channels, they are real blockers. They are real power blockers. They interact with the inner part of the ion channels, and one of the mechanisms related to this power blocker is based on a functional diet on that side chain of a lysine interacts in the selective filter of the channel, and the tyrosine or another aromatic residue interacts in the outer part, stabilizing this interaction, and this promotes this stop the flux of ions. For this presentation, I choose to talk about the 3 principal toxins that were described in the venom of the Titus scorpions, and I would like to start in talking about the TS1-like toxins. As you could see here, you have the TST1, TS1, TB1, TF1, and TT1G. These are from scorpions: Titus stigmurus, Titus cerulatus, Titus baiensis, Titus fasciolatus, and Titus triviutatus. And again, Titus fasciolatus. It's important to observe that these toxins has a really high identity between them. Using the TS1 as reference, the maximum difference is in 93%. And when we look the alignment, it's possible to observe that the most variability of these toxins it's observed in the alpha helix, as could be seen here, as could be also been seen here in this 3D representation of the TS1 toxins. It was highlighted the regions of the high variability. And for me, it's really interesting when you compare 3 toxins: the TS1, TST1, and TF1A in the same concentration at the channel NAV 1.2. TS1 on the channel NAV 1.2 promotes a slight. First, it's important to explain what this type of graph. This type of graph is a graph that you can simultaneously analyze the activation of the channel in this ascending curve, and also it's possible to observe the steady state inactivation of the channel. Beta toxins interacting in the domain 2 promotes the stabilization of the voltage of the domain 2 in the up position. This promotes an early open of the channel in voltage, in more negative voltage. As could be seen here, TS1 promotes this early open, but TS2 that has just few difference promotes a stronger shift in this open probability, making the channel open really early. And the other toxin, the TF1, promotes also a shift stronger than TS1, but not stronger as the TST1. And it's important to see that this difference is just in the alpha helix, and sometimes we have some conservative modifications in the C terminal. And also, for me, it's really interesting that all the toxins of the TS1-like toxins are amidated, C-terminal amidated. And GKK is a signal for the post-translational modification that cuts and amidated the cysteines. The next group that I'd like to present is the TS3-like toxins. And this, for me, it's really interesting because it's most recently in 2018, for the first time, was cryo-EM structures describe the interface of interaction of alpha toxin and in the sodium channels. And these interactions are main in the beta 3, the loop between beta 2 and beta 2 strands, and also the other two sites of interaction is the reverse turn and the C-terminal segment. And when we observe the alignment of these toxins from Titus cerulatus, Titus stigmurus, Titus fasciolatus, and the two toxins from Titus baiense, it's possible to observe that also the variability comes from the alpha helix, but also comes from the beta 2 and beta 3 loop, and some differentiations in the C-terminal segment. And look as a, and just to exemplify the effect of alpha toxin, I select to choose this current that we can demonstrate, that I can demonstrate in black the control condition of a current NAV 1.7 current, and after the application of TS3 at 200 nanomolars, we have a higher amplitude of current, but we have the classical effect of alpha toxins. It's the delay in the fast activation. So, to study how this few difference that we found on TS3 and TST3, that is some difference on the loop beta 2, beta 3, exactly this part, and the difference between the C-terminal segment, because in TS3 we have amidated serine, but in TST3 this serine is not amidated. So we try to understand how this difference could affect the toxin interaction to the channels. So, in this paper, we use two different approaches to analyze this problem. First, we use the classical experimental approach, and also we use some computational analysis to analyze if these three differences on the sequence between TS3 and TS3 could affect the interaction. Paying attention on the experimental approach, it's possible to observe that TS3 and TS3 promote similar displacement of the steady-state activation, both enhancing the stabilization of the inactivated states, and also promotes the same degree of non-inactivation current, and also promotes similar degrees of the delay in the fast activation. The second approach related to the computational analysis, we analyze how these toxins interact in how these three differentials could affect the stabilization of the toxin in the voltage sensors in the up state, in the down state. And we observe that the toxin has preference to interact with the toxin in the down state, as could be seen with this negative values of delta G. And also we calculated for all the toxins. Oh, sorry, I changed. So returning, we calculated the difference between the two states, the up states and down states, and observed that toxin really interacts better in the down state, what was expected for an alpha toxin. And when compare the values between TS3 and TS3 and TST3 interactions, they are kind of similar. So in the final, we observed that these three modifications didn't change how these two toxins affected NAV 1.4 channel. But to take advantage of this new approach of screening, we use also the classical experimental one recording this current on patch clamp. And we observe that TS3 has a high spectrum of activity on NAV 1.1, 1.2, 1.3, 1.4, also demonstrating energies related to a better stabilization of the voltage sensors in the down position. And also we have made this for NAV 1.5, 1.6, 1.7. The only channel that our computational analysis didn't work well was for NAV 1.6, that we have a signal that that demonstrate that this channel is better, the toxin interacts better with the voltage sensor in the up position instead of the down position. And also we use this data to try to rank the selectivity of these toxins between to rank this toxin, the affinity of the toxin between NAV 1.1 and NAV 1.7. And was observed using the difference of the time constant of the fast activation, that the TS3-like toxins has more affinity to NAV 1.7, followed by NAV 1.5, NAV 1.2, 1.3, and 1.6. And in the last ones is 1.1 and 1.4. And this is really interesting because in the previous studies, TS3 was just tested on NAV 1.4. And our recent study demonstrated that this type of toxin, that the TS3 similar toxins, has more selectivity to NAV 1.7. This group of toxins, the TS2-like toxins, is, for me, it's really, really interesting because we have two different toxins in this group. The first one is the TF2. TF2 is a toxin that was described as a high selective toxin to modulated NAV 1.3 with beta factors, as could be seen. It promotes enhancement of currents in low voltage, as could be seen control in black and in red for the toxin condition. So the toxin promotes the early open of the channel, as also could be seen here in this graph. But in the same family of toxins, the TS2-like toxins, we have TS2. And TS2, for me, it's really it's different. TS2 also has some beta factors that could be seen in this graph, in this part, using black as control and blue circles for our toxin condition. It's possible to observe that the toxin shift the activation of the channels to open earlier. But meanwhile, these toxins also promotes alpha activity. So in the same toxins, in the same group, two toxins with just 5% of difference in identity promotes two completely different activities. One is a beta activity toxin, and the other, I don't know how to explain, is beta toxins with alpha activity or alpha toxins with beta. I don't know. And some studies pay attention of this high selectivity of the TF3 to NAV 1.3 the penetration of the activity of TF2 on NAV 1.3, and one really interesting thing is that NAV 1.3 is upregulated after nerve injury and could be one of the targets to treat neuropathic pain. But one problem related to beta toxins is that beta toxins has a dual mode of effects. Beta toxins is depressant, as you could see here. It reduces the amplitude of the current, but in the same time, it's excitatory, that because promotes an early open of the channel. So to treat any disease that we want to silence the specific channel, it's really important to have a toxin that just block without modulation. And for this reason, this study make one mutant named is TF2 with a modification of serine 14 to arginine. It's a mutation known to reduce the excitability effect of beta toxins, as could be seen here in this graph comparing to TF2. The modified TS2 reduce this capability of shift in the activation of the channel, but and also maintain the depressant effect to reduce currents. But one problem that happens when you do this is you lost sensibility of the chain of the toxins. It's important to mention that TF2 was tested at 1 micromolar and TF2 modified was tested at 10 micromolar. And to prove that this type of toxins could be used for neuropathic pain, some experiments on mice, some intraplantar injections of TF2 and TF2 modified and specific blockers of the NaV 1.3 demonstrated that the TF2 promotes the white type TF2 promotes some responses related to pain, and also when they use the TF2 modifier, this response of pain were reduced. This is probably due the suppression of this excitatory modes of the TF2. And to analyze if the TF2 and TF2 modified could comparing for the same binding site, they put both together and observe that TF2 probably has more affinity for the NaV 1.3, and the signals of pain were higher again. And after that, they try to understand if the use of TF2 associated with a blocker of NaV 1.3 could decrease the signal of pain. But what this data probably demonstrates that the TF2 probably has a better affinity for this target, and this could also enhance pain, could lead to pain. Also, meanwhile, with the presence of a specific blocker. In this study, they also evaluated how the PA of the mice, the inflammation of the mice PA will be modulated by the TF2. And they observed that the TF2 and TF2 modified also promotes the swelling of the body, and also this is possible related to another target, not related to NaV 1.3, and that's the reason why this toxin didn't go to other next researchers, because they demonstrate that the TF2 is related to some inflammation process, not related to NaV 1.3 activation. And I have talked a lot about the toxicity on sodium channels, but we have also the toxicity on potassium channel, the KTX. And we have recently described a new toxins, alpha KTX 4.9, described from the venom of the Titus fasciolatus. It's a new member of the family 4. It's a family known to be a Titus family. Just recently, another toxin from other genre was described in this family. It was the CM39 from Centroides margaritatus, but all the other toxins in this family are Titus toxins. And these toxins, the TF5 was described as a power blocker of Kv 1.2 and Kv 1.3 with AC50 of 15 for Kv 1.2 and AC50 of 116 for NaV 1.3. When we are studying toxins activity on potassium channels, we are always looking for specific blockers of Kv 1.3 channels, a diverse of therapeutic applications that could be used. And for this moment that we try to study this toxin, we were thinking that we were in between two types of toxins. Highlighting green here, we have in the position 29 some toxins with methionine and other toxins with isoleucine, in some cases of threonine. Some studies reported that toxins that has the methionine in the position 29 are related to toxins with more activity on channels, on Kv 1.2 channels, and toxins with the isoleucine are more active in 1.2. So for this study, we are expecting to see a toxin with higher selectivity for Kv 1.3, but it was not the case. And why it's important to study targets for study Kv 1.3 as a target for neurotoxins? I think this is the most classic explanation, is the that KV1.3 regulates T cell activity and differentiation. How this works? When an antigen-present cell presents the antigen to a T cell, this promotes some cascade of events that leads to the release of the calcium-free that exists in the reticular endoplasmatic. And this calcium is a signal to activate a channel named CRAC. CRAC is calcium release activation channel. It's a channel with the signal of calcium release allows the open of calcium. And this despolarizes the membrane. And after despolarizing the membrane, the potassium 1.3 is activated. It's important that the signal of the enhancement of calcium intracellular, it's a signal for proliferation, activation, and also for more expression of KV1.3. When KV1.3 is blocked, this whole cycle is blocked because we have the activation, this despolarize the membrane, but with the channel 1.3 blocked, we don't have the repolarization of that membrane, so this cannot repeat. And this is a therapeutic approach to treat some autoimmune disease related to T cell activity. Nowadays, we have two specific peptides with high activity, with high specificity to KV1.3. We have the promised one, VM24. It's a toxin isolated from Biovis mexicanus, a scorpion that is found, as the name saying, in Mexico. And also has a KD of 2.9 picomolar. It's a promising one, and nowadays it's not on clinical trials, but has another studies demonstrating some modification in the sequence to promote more affinity to KV1.3 and reduce some others of target activities. And nowadays we have a toxin that was described for a venom of cyanemone, and now it has a commercial name of dalazantide, and it was used in clinical trials for treatment of psoriasis. And more recently, in a paper which was published 1 or 2 months ago, it was demonstrated that TS6, a toxin isolated of Titus cerulatus, or identical toxins isolated from Titus baiense, the alpha KTX12, both it's active on 1.3 and 1.2, promote signals of neuroprotection. So that's that I have for today, and I also need to thank you for the group of Elizabeth Ferroni. It's she has been my tutor for more than 10 years, and I'm really grateful for all the things that I learned with her. I need to thank the University of Brasília, the program that I've done all my post-graduations, the lab, Neuropharma Lab, the laboratory of computational and theoretical biology that make all the analysis related to computational analysis that I demonstrated for TS3 and TST3, and also the founders from CAPS, NPQ, and FAPDEF. Thanks so much.
Thank you very much, Dr. Tibery. It's a great talk. So, anyone has questions? Professor Marilena?
Great talk, Diogo.
Thanks so much.
We know that most of these toxins are promiscuous, as you showed. About your work, which of these toxins do you think are more promising to arrive to have a drug, pharmacological drug?
Yeah, most of these toxins are really promiscuous, interacting in most of the ion chains, as I demonstrated before, the TS3 and TST3 interacting with all the chains from NaV1.1 to NaV1.7. For the toxins described from scorpion venoms, active on sodium, the most selective one is TF2. But how I say, it's a gate modifying toxin. It so has activity as a inhibiting toxin and a excitatory toxin in the same time. So I don't understand the toxic activity on sodium channels Oh, this is better. I don't see the alpha toxin, the scorpion toxins as a good, the scorpion toxicity on sodium channels a good promising model for treat some disease.
But perhaps, as you show, if you can change some residues, amino acids, you can arrive at a specific drug, because you show that when you change some residues, you have the different activity. So perhaps it's a good approach for the future. Don't know, but it's complicated because the structure is very complex. But thank you, very nice presentation.
All right. Anyone? More question? No? Thanks to Dr. Tibery. Great job. Let's move on to our next guest. I think she's online now.